Okay, so hello, very nice to be here. Thank you for the invitation. So I work not far from here at AgroParisTech campus on the same plateau. So I'm a soil scientist, and my current research is on carbon storage in agricultural soils, and I'm specifically interested in fine-scale processes at the level of the microbial habitat. But my main activities in the last years has been to coordinate a wide European research program on climate smart and sustainable management of agricultural soil, the AJP soil. So today, yes, I'd like to talk about the plant's contribution to carbon sequestration in soil, and I have prepared this presentation with one of my young colleagues, Frédéric Ries. He could not be here today because he had long planned holidays. Okay, so soils are essential. Well, of course, say that because they provide many ecosystem services, well, support of food production, regulation of the water cycle, contribution to mitigating climate change, recycling nutrients, supporting biodiversity. And we define soil health as the actual capacity of a soil to provide ecosystem services. Soil degradation, of course, is when soil health is not ensured, when they do not provide all the ecosystem services they could. And the current situation is quite preoccupating because, for example, for Europe, the GRC estimates now that 62% of European soils are being degraded, are not healthy anymore. And organic matter is really at the center of soil health because it contributes to primary productivity, to biodiversity and habitat provision, water purification and regulation, nutrient cycling, and climate regulation. So it's one of the main and the main indicator of soil health if you want to have only one. So soil organic matter is closely related to soil health, and it is also crucial for one of the challenges of our century, that is mitigating climate change. Here you have one of the latest carbon balance of our planet with different pools and fluxes, and soils correspond to the largest terrestrial pools. If you consider the amount of carbon that is in the upper layer of soils, 40 centimeters worldwide, you have 800 gigatons of carbon. Now, if these 800 gigatons per year would be decreased at a rate of 4 per mil per year, 0.4%, because of mismanagement, because of changes in land use, it would actually double the net flux of carbon from our planet to the atmosphere. And on the opposite, if it was possible to increase this global carbon pool in soils by 4 per mil per year, it would offset this flux. And this was the idea at the onset of the 4 per mil initiative that was launched by France at the COP 21. In fact, it's a very ambitious goal, but still, to give you an order of magnitude, soils are extremely important for carbon, for the carbon balance of our planet. So what I will do is I'll go first rapidly through what is organic matter, and then address processes and maybe orders of magnitude of the contributions of plant to soil carbon sequestration, focus on resource deposition, and then go through management of plants for carbon sequestration. So soil organic matter is the result of plant inputs to soil being processed by microorganisms and by soil biota, and it ends with mineralization, knowing that other fluxes or the outputs are erosion or leaching, but you see the relative size of my arrows. So it's something that is constantly being renewed, and it's a mixture, an awful mixture of plant debris, of live biota, dead biota, molecules that are free, that are associated to minerals. And it's not only, half of it is carbon, but there's not only carbon. So the mineralization releases a number of elements that are nutrients for plant. So you see from the climate change mitigation perspective, you would like to hoard soil carbon to really sequester it and that it doesn't move anymore. And from a soil functioning perspective and a plant perspective, you need this recycling. You need the organic matter to be used by soil microorganisms. And this constant biodegradation and renewal takes time. And in fact, mean residence time of an organic molecule in soil goes from 1 hour to millennia, with an average residence time in our temperate areas of 50 years. And we usually separate this pool of carbon into different pools with different residence times. You see months, decade, century. This heterogeneity of residence time and transformation speeds results from the diversity of the controlling factors. And we will see some of them later. Now, what is plants' contribution to soil organic matter and soil organic carbon? I rapidly assembled a few numbers from different sources because I have to give gross estimates. Actually, most of soil organic carbon derives from plants, because you see here numbers I found for exogenous organic matter input, manure, compost. Well, these are not applied on most cropland right now, but when they are applied, average numbers would give you that 1/4 comes from that, more than half from plant. And what I added is the biomass of soil fauna and the biomass of soil microorganisms. So if we would assume that they all die and go then to feed inert soil organic matter, these would be numbers. Of course, it's much less than that. So the vast majority of soil organic carbon really comes from plant biomass. Another point I wanted to make is that plant biochemical quality does matter for organic carbon storage, also the stoichiometry between elements. But regarding biochemical quality, in the short term, several months, a year, the biochemical quality has been shown extensively to be a fairly good predictor of how much is left from a biomass source. For example, this is one example of one of the indexes that is used now by professional labs, is that you can predict the proportion, the ISMO, is the proportion of carbon from a biomass that you bring to soil that remains after 1 year. You see, it is proportional to the amount of soluble in the biomass, to the amount of cellulose and lignins. So it works, but if you consider longer timescales, that is decades, centuries, we have, well, the scientific community has found, well, I would say since the last 20 years or 15 years, that it's not possible to make any strong correlation between the chemical nature of the soil organic matter and its persistence. So what it means? So from that means that there has been changing paradigms. So what it means basically is that if you consider longer timescales, there are other processes that just override the effect of biochemical nature of the plant biomass of the soil organic matter, that just erase it. So the initial idea was that persistence was really linked to breakout sequence, intrinsic chemical composition. What we know now is that, yes, char are very good candidates for long-term persistence, but nearly that's all. But there are other processes that organic matter may be stabilized in soil by interaction with organomineral interactions, complexation with cations, adsorption to clay minerals that really protects it against biodegradation, but also by entrapment in small pores in soil, in aggregates, where either the physical conditions, availability of oxygen, or the accessibility to microbes are not proper for biodegradation. So these processes really overcome the effect of the biochemical nature of the biomass inputs. And actually, there has been a set of changing paradigms in this area. And now the vision of soil scientists is more that persistent organic matter is, of course, a little bit inherited plant material, well, partly inherited plant material, plant molecules, but it would be mainly plant material, plant molecules that have been assimilated by microbes who died. So it would be essentially microbial necromass. So, you know, it changes the vision. Another point I would like to make is that roots do matter for soil organic carbon storage. And what has been demonstrated using isotopic tracing or isotopic labeling with C13 is that the yield in soil organic carbon, you see my color code, yellow for the roots, the below ground biomass, green for the above ground biomass, the yield in soil organic carbon is more than 2 times higher when the biomass is below ground biomass compared to above ground biomass. So in a soil carbon sequestration perspective, have to care about the below ground biomass. Why? Why? Well, roots tend to incorporate carbon in many different locations in soil at the same time, access to micropores, access to mineral surfaces. And then, of course, one of the hypotheses was that suberin is a candidate for being persistent. Explains partly not that important. But it would be essentially that root rhizole deposits would be absorbed to soil minerals and would be trapped into small pores where they would be inaccessible to microorganisms. And again, it's really the fact that roots deposit, rhizole deposit in many different locations, really deep in the soil architecture. So what are these rhizole deposits? So this is the area of work of my colleague Frédéric. There are different rhizole deposits that are emitted in soil. First, there is what is well known, best known, I would say, is the exudation of soluble compounds, that is diffusion, facilitated diffusion through a cell membrane or active transport for some compounds. And to quantify this, the experimental approach usually is to work with hydroponics and analyze the solution, extract them from the solution of the hydroponics. In soils, we don't know how to do it. Second is the secretion of mucilage. So high molecular weight polysaccharides transported outside of the cell by vesicles. And here, the experimental approach is, well, in soils, taking out the roots and really sucking up the material. Well, in cases like that, it's fairly easy. You can recover a fair amount of material. And otherwise, to grow the plant in sand or assemblages of glass beads, and then recover this suspension, centrifuge, etc. In soils, really tricky to do it. Third compartment, third fraction is the sloughing of cap cells. And here the methods are to enumerate and also to apply flow cytometry or microscopy flow cytometry to recover those cap cells that have been slaughtered. And a fourth fraction are volatile organic compounds that would be actively or passively emitted in relation to cell aggregation. For that, at the moment, it's essentially, well, handmade devices to try to quantify them. And yes, for example, yes, if you are interested, one of the PhD students of Frédéric is going to defend her viva early May about quantifying volatile organic compounds emitted by root systems. And she developed a whole setup to do that. So 4 different fractions of rhizole deposit. And actually, the situation is that we do not have methods to quantify that. That's a real difficulty. What we have in the literature well, and from different works, are global estimates of rhizome deposition. Here, for example, are numbers of total carbon inputs by gross deposition in cereals, in perennial grasses, where you see GDP, well, shoot allocation, then allocation to roots, and then the gross rhizome deposition here. So numbers are 5 to 15, maybe 20% of the photosynthesized carbon. So it's not negligible, not at all. So yes, it would correspond to a few tons of carbon per hectare. Well, actually, it's more or less the same amounts at manure application rates when manure is applied to soils. So yeah, it's really an issue to be able to quantify, but we know it's an important, it's non-negligible fraction. And if I go back to, yes, what is the carbon cycle related to this? So we're interested in rhizome deposits. Of course, there is photosynthesis and respiration, allocation of carbon by the plant to below ground, respiration of the roots that must be accounted for. Part of the roots die and they contribute to soil organic matter. Part is exuded, is produced as rhizome deposits, which can be stabilized in soil and persist as soil organic matter, or can be assimilated by microorganisms which respire. And yes, there are also of course, use of, I would say, we can say indigenous, well, existing soil organic matter and soil organic carbon by microorganisms, and their necromass will feed this pool. So here, so this is typically what we would like to be able to document, to put numbers in our lab, but there are really analytical problems. Rhizome deposits are produced in low numbers that do not persist for long because they are rapidly used by microorganisms. So it's a very small and transient pool. It's a pool that has the same chemical nature as the roots themselves, as the microorganisms biomass exudate. So we do not have any criteria, biochemical criteria to distinguish. And yes, differentiating the different sources of CO2 requires to use isotopes, labeling or natural abundance. There are also ecological problems associated. It is thought, in some cases shown, that rhizome deposition is less in the model systems we tend to use, hydroponics, glass beads, sand, than in real soils. And also conceptual problems. What to do with mycorrhiza? We consider that they're part of rhizome deposits of the wood systems. And what is in fact the difference between dead roots and rhizome deposits? It's difficult to put the limits. So many challenges that are being addressed, and that are addressed in particular by Frédéric now. But yes, we think it's really important, given that rhizome deposits are good candidates for long-term stabilization of organic carbon. It is really important to decipher this cycle and to document it with numbers. Also, it's in fact a bit more complicated. There are other processes. For example, microorganisms, when they can assimilate easily usable compounds, organic compounds, they overproduce extracellular enzymes, which then biodegrade, mineralize resident organic matter. We call that priming the priming effect, and it causes an additional flux of carbon to the atmosphere. So roots both contribute to stabilization and sequestration of carbon, but also to its destabilization. And it's so regarding roots, it's called the rhizosphere priming effect. And I wanted to show you one example of quantification for different plant types, grasses, forbs, legumes, according to phenological stage. what you can see, well, RPO is a rhizosphere priming effect, is that it nearly doubles the net flux of CO2 from the indigenous soil organic matter. So it's really has to be accounted for. So far, it seems that the balance is always that there's more carbon sequestered from roots in the midterm than destabilized, but the balance needs to be established. Other interactions is that roots favor aggregation. Well, you very likely notice that when you take a plant, there's a lot of soil that comes attached to the roots. These are, they tend to stick the soil particles together to create aggregates in which soil organic matter may be protected. So it may have a negative influence on biodegradation of organic matter. So this is an indirect effect on soil organic matter and soil organic carbon stabilization. So different interacting processes. So this was processes, quantification and processes. Now, a few insights about managing soil organic carbon stocks via plants. And I will start with a general overview of how to protect soil organic carbon stocks, how to increase them with a simplified carbon cycle at the scale of an agricultural plot. There are a number of levers, actually. There's no magic solution. There are a wide set of solutions. Increasing photosynthesis in time with intercrops in space, and also with increasing the proportion of temporary layers in rotations, increasing photosynthesis in space by grassing interrows in orchards and vineyards, by implementing agroforestry, banning, reducing fires that are just a net loss of carbon from the agroecosystem, playing on the balance between exports and return of crop residues, importing organic matter from elsewhere, manure from other plots, from breeding and or compost from cities, for example. All these, and also playing on the grazing versus mowing, the density of the cattle, all that affects the inputs. All these affect the inputs of organic carbon to soil. And now the evolution of organic carbon stocks, of course, depends on the balance between the inputs and the outputs. And so is it possible to reduce erosion? Well, levers are terraces, well, you know, at the infrastructures at the landscape scale, but also at the plot scale, agroforestry, conservation agriculture. There is no tillage with permanent plant coverage. And is it possible to reduce mineralization of organic carbon in soil? Partly by reducing soil disturbance in no-till and conservation agriculture system. Inputs or outputs? Finally, what is most efficient to play with? And here I have chosen to show you a few results from a long-term field experiment that is fairly close to here. It's actually located in Versailles, where since 98 are compared different cropping systems. It's in cereal culture, wheat 1/2, conventional productive agriculture, well, you know what it is, but there's also a system with low input, so reduced tillage, reduced fertilization, reduced pesticides use. There's a system under conservation agriculture, so it's based on the low input system, plus there is no tillage, and there's a permanent plant cover. That is alfalfa right now. And the fourth system is organic agriculture, so no mineral fertilization, no pesticides, well, no synthetic pesticides, according to the regulations of organic agriculture. And you see that the yields vary quite a lot accordingly. What we did in this long-term experiment is that we measured organic carbon stocks. So I'm sorry, I will not show you the latest results, but the ones from 2014, but well, the trends are exactly the same, except that it's more pronounced. We found that none of the system had any effect on carbon stocks, except the conservation agriculture one. And actually, when we calculate the annual increase of organic carbon stocks, well, for you, it will not mean anything, but for us, 0.5 ton per year and per hectare of increase is really big. It's really big, so very efficient. Now, what explains it? So what we did was to quantify using the records of the techniques and rotations and measurements that were done of yield to quantify the organic input. You see the cash crop is very variable. Cash crop input is very variable. That's in relation with the yield numbers I showed you. But the conservation agriculture is actually the system that has most input, even though its cash crop input is less than in the low input system or in the conventional system. You see, because if you consider the inputs by the cover crop, by the permanent cover crop, the alfalfa. And what we did also was to separate, and well, for that we had to use functions, allometric functions. We tried to estimate above ground and below ground input. We didn't measure, we tried to estimate. And we found that in the conservation agricultural system, below ground inputs were proportionally higher than in the conventional system and in others. So increased inputs, 1/3 in addition, increased below ground inputs. So increased input for sure. And then we wanted to assess whether the outputs were increased. And we, in the lab, well, we couldn't measure it in the field, but in the lab, we found absolutely no difference in mineralization rates. We complemented that with quite a lot of modeling to test hypotheses. But well, the result is that here, the additional carbon storage depends on increased inputs. And actually, we found we did more or less the same set of measurements in an agroforestry system over 15 years, found the same result. And the other results in the literature recently tend to favor that to increase organic carbon stocks, the efficient lever is to increase inputs. And inputs are essentially plant inputs. So what could matter elsewhere? What could be managed? Plant diversity. Plant diversity, well, there's a lot of questioning in our community on that. There are not that many measurements. There's one paper that had quite a lot of impact in the Jena experiment. It is an experiment on the grassland where they manipulate plant diversity. You see plant species richness with quite an amazing range of plant species richness. And they measured over 8 years the variation of organic carbon stocks, contents and stocks, and you see an increase. So apparently, well, from this study, plant diversity, increasing plant diversity increases organic carbon stocks. What can be behind? Niche, probably niche complementarity in root systems, so that the soil is more used, explored by roots. But well, this really needs to be sustained and complemented by information in crop soils, and in the European Joint Program I'm coordinating, the HAP soil. So far, we have well, it's not yet published, but very variable results according to pedoclimatic conditions, to rotations. So no simple solution. We need to analyze, all the different teams and projects who worked on that really need to analyze in detail the results. But well, that's a thread to follow. Another variable trait is rooting depth. So here is a result that has, it's not directly about roots, a meta-analysis of carbon age distribution in different grassland and forest soil. And you see that the distribution of ages is really changes with depth. And actually the median, if you look at the median at 20 centimeters is about 50 years, the median at 50 centimeters is about 300 years. So in a carbon sequestration perspective, where you want the carbon to stay there as long as possible, you have to go deep. We have to go deep. That means maybe implementing root systems.
He's in the meeting in Pascal, Institute Pascal.
Someone could switch off. Hannes, could you switch off your microphone?
Okay, doesn't. Okay, so yes, go deep. And again, why? Why does it persist longer? Well, quite simply, because there's less oxygen at depth. And there's less biological activity because the oxygen concentrations are less favorable to decomposing activity of microorganisms. A question that comes very often to us is that, well, okay, you're a nice soil scientist, you want us to breed plants, to put more carbon in the soil, to develop larger root system, deeper root system, but it compromises, it's really bad for, really poor for yields. And in the AJP soil, one of the team performed a meta-analysis of field experiments with different varieties of a series of crops that you have here. And what is represented here is the biomass, the root biomass change in percentage compared to the median of the results for this crop. And in the y-axis, the yield change, same type of representation. And you see that, of course, there are situations here in orange where increasing root biomass was either has no effect or is detrimental to yields, but there are also positive situations. So yes, we can think of optimized genotype selection as a win-win option, both to put more carbon in the soil and to maintain or even increase yields. But I'm curious about you probably have other data on that. So the question of trade-offs is really important. Trade-offs between carbon sequestration and other functions expected, and productivity mainly, but other functions also expected from soils. And this is quite difficult to assess experimentally. There's clearly not enough data at the moment. There are a few modeling studies, and I want to present one of them, again, done in the AJP soil, where colleagues used a crop model that they enriched with an exudation function, that they enriched with accounting for soil structure, with accounting for several soil physical properties. And then they worked with wheat and defined ideotypes by changing model parameter values. So they had, so they played on the elongation of roots, density of root hair, the branching of roots in the architecture part of the model. So to have increased below ground allocation of organic matter via the root systems, to have deeper roots, to have more effective roots for water uptake. So that's, if I remember well, it was essentially through root hair and fine roots, and a combination of all three. And they could not validate for all outputs of the model, but they validated for yields, harvest index, and wood to shoot ratio in long-term experiments in Sweden. What they found is that increased below ground allocation to root and deeper or more effective roots for water uptake slightly increased yields, slightly increased soil organic matter contents. So positive synergies, I would say. Combination of all three increased definitely increased yields and soil organic matter content. Now, regarding other functions, runoff, there was synergy, clearly reducing runoff in the three simulations. But regarding the recharge of the stocks of water under soil, there was a trade-off. That can be expected, more transpiration by the roots. So yeah, they found an additive effect of phenotypes that crop variety selection for improved root traits actually could make a good contribution to soil organic carbon stocks and yields. And there were trade-offs and synergies. Synergies for erosion mitigation, but maybe trade-offs for the water balance. So yeah, models are definitely a tool that we can use to continue. But there's a need also to, of course, to calibrate and validate with more experimental data. So I'm coming to my conclusion. So one of the things I would like, yes, to end it is that really carbon sequestration, very often the idea is what is important is sequester carbon and that it doesn't move anymore, doesn't decompose. No, we do need both to use carbon for soil fertility, for biological activity, and to hoard it for climate change mitigation. And it is possible to have both at the same time. It's not either or, it's possible. It's a number of management options. Roots and rhizole deposits do matter for carbon sequestration in soils. They are changing paradigms. So maybe the idea to retain is that what drives decomposition is not only the chemical nature, but it's the environment, the clays that are there, the soil structure, the physiology of microorganisms, the physical and biological environment. And there are challenges and open questions. Well, I've showed you that for rhizole deposition, quantification is really, really a problem. To quantify also carbon and nitrogen fluxes in the rhizosphere. There are open questions and challenges regarding plant diversity effects, root trace, what are the positive root trace for carbon sequestration? And of course, challenges also to go from process understanding to management options that could be co-constructed and implemented in the field. And to finish, I would like to tell you who we are. So we are on the plateau of Saclay. We are a research unit named EcoSys, where there are two teams. One, Solitox, it's soil science and ecotoxicology in soils. I'm in this team. And the other one is EcoFi, ecophysiologists essentially. And they are interested, if you are interested in ecophysiology and modeling, yes, I prepared that. So please come and meet. You can come and meet us. You're most welcome. And I didn't dare to show you one of the results of Frédéric, although it's really spectacular, because I thought my computer would not handle, but you can find it on BioX3, is that he's modeling the evolution, the temporal evolution of root architecture and rhizole deposition. And on that, I would like to thank you for your attention.